So zebrafish is actually a tiny little animal that doesn't look like much. Like the adults are very small, and I think maybe you're gonna show some. And it became very popular about 20 years ago. The eggs were transparent, and so it became a very good model for studying development, because you can basically see how a little fish is developing in a very short time. So in 24 hours, you basically have the basic body plan of a vertebrate, and you see how the brain, the spinal cord, the different organs come up, and you can follow that by simply putting your larvae or your embryo under the microscope. So
the relevance of our work is, since there's been a lot of focus in the legalization of medical marijuana use, there's been increased interest in knowing what's marijuana doing to the brain.
My research is about the regeneration of a specific tissue that is a sensory tissue, the hair cells. The main aim is to understand the mechanism of the regeneration of these tissues.
In my research, I'm focused on using the CRISPR-Cas9 technique. And basically this technique has been revolutionary, most people will say, in the genetic field. And this technique will allow us to be specific, to target specific genes and actually edit these genes in order to get mutations of any gene of our interest.
My main focus in the lab is, I would say, hair cell regeneration. So it's a regeneration of a sensory epithelium. And so what is this? So it's basically in our inner ear. So what you're using right now to listen to me, okay, is you're having, it's a pretty complex 3D structure that is deeply buried inside of the brain. But really it boils down, the hearing boils down to these sensory patches. So they're very discrete patches in the ear, and the hearing basically is in the cochlea. So you're having this sensory epithelium that is made of what is called a mechanoreceptor, which are these hair cells.
Since the zebrafish is a vertebrate animal model, we can also do complementary research along with the mice to continue doing behaviors and identifying the role of these receptors in different behavioral tasks. For example, anxiety, learning, social behavior, other cognitive behaviors, and neurological disorders.
We've been able to address and target genes involved in cardiac tissue. Like, for example, there's the myosin-binding protein, C gene, which is involved in the structure of the cardiac tissue. And it's known to be a big player in some diseases like hypercardiomyopathies. So once we have our meeting, we're going to be able to understand what's happening when this gene is not functioning properly.
The zebrafish has really gained a lot of momentum in the past year, and it's becoming like really a major player in basic science, but I think it will also be true in industry. One of my roles when I came here was really to get this animal model going and to offer the opportunity to the students here to be able to do top-notch research with this really nice animal model. And I think this will open doors for them, not only in academia, but also in industry.